Welcome back, everyone. I'm delighted to introduce our very first plenary presentation session of the meeting on variants and functions. This session will be moderated by Matt Hurles, who is the head of human genetics and senior group leader at the Wellcome Sanger Institute, and also a member of ICDA's organizing committee. Please remember to ask and upvote your questions in Slido and continue the discussion in our Slack channel. All right, Matt, take it away.
Great. Well, thank you very much, Rachel, and welcome everyone to this first plenary session. So we've got an excellent set of 4 talks for you, and it really spans the range of activities in this area within ICDA. So we'll be hearing about gaining a fuller picture on genetic diversity and diverse populations. We'll be hearing about cellular genetic technologies, probing function in disease-relevant genes. We'll be hearing about single-cell RNA sequencing, and its ability to understand the functions of genetic associations. And we'll also be hearing about some sophisticated data analytic methods that improve our ability to interpret genetic variation that is hard to interpret currently. So as Rachel mentioned, please ask the questions through Slido and upweight them as well. Now, just to note that the second of our talks from Anna Gloyn is pre-recorded, and so there won't be a Q&A through Slido, but please pose questions to Anna through the Slack channel. Okay, so without further ado, it's a great pleasure to welcome our first speaker today, who's Eimear Kenny. She's a statistical and population geneticist. She's founding director of the Institute for Genomic Health and associate professor of medicine and genetics at Mount Sinai. And she's going to be talking to us today about risk prediction in an era of massive scale genomic data. So Eimear, over to you.
Thank you, Matt, and thank you, Eric and the other organizers and Cecilia for inviting me to speak today. This talk is, of course, tweetable with the hashtag for the conference is ICDAbio 2020. So my disclosures. So I wanted to talk about today, you'll notice my talk title is a little bit different. I really wanted to focus on questions at the interface of where we are in the field of human genomics with massive scale data and how that plays out across, particularly across risk prediction across diverse populations. So around this time last year at the ABGT conference, Illumina announced that 150 petabytes of data had come off their sequencers that year. In that same year, 2019, YouTube announced that they had generated 93 petabytes of social media data, and comparing that also to other large research efforts in different fields. For example, what's going on in CERN in the Large Hadron Collider, they produced 25 petabytes of data. So I think it's fair to say that genomic technology is really creating some of our biggest databases of knowledge. In particularly in human genomics, we're in an era now where we have assayed genomes of hundreds of millions of humans on the planet, which is now getting to be quite an appreciable slice of humanity. And of course, that data itself can be linked to cellular tissue, organismal, and outcomes pertaining to health, pertaining to disease, pertaining to social science, anthropometric traits, biomarkers, and this is really a very exciting time, I think, in the field of genomics, because we are living in a very data-rich time. It is particularly interesting to think about the question of how we use genomic data for risk prediction, or even protection prediction for different diseases and traits in humans. This is something that has been done in animals and plants for a long time, but it's really only with these sample sizes in humans that we can finally get to the tipping point where these data can be useful for predicting risk. But of course, as we build these big databases, we do also have to acknowledge some of the biases currently therein. This is one of them that is the focus of research in my group, and that is that genomic databases are failing on diversity. So this is data from the GWAS catalog. It's a fantastic database. I'm not picking on it. You'll see the same picture in other databases. But in the GWAS database, 80% of participants are of European ancestry, probably more specifically of Northwestern European ancestry, and there is underrepresentation or even lack of representation for many other populations around the world. So a couple of years ago, in work led by Alicia Martin, as she was a graduate student, we asked the question, well, what impact does this bias in diversity have on our ability to use these databases for prediction? And long story short, what we found across a few different traits at the time was that if you take a PRS score that's generated predominantly from European ancestry populations, and then you use it naively to predict outcomes out of sample and out of European populations, then you will see a mean shift in the distribution in different populations. And it is actually very variable depending on which trait or outcome that you were looking at. And at the time that we published this paper, we deduced that neutral human evolution was sufficient to explain these differences. That is, differences in allele frequency and LD structure across populations were sufficient. And I think in the short time since we published that paper and today, we actually have learned an awful lot about what's going on underlying these differences. And it's certainly not as simple as just LD and allele frequency. And here's one example of drivers of differences in prediction across populations. This is a missense variant in the 2IRP1 gene that underlies 50% of variance in hair color in Melanesian populations from the Solomon Islands. And that 25% frequency gene by itself is a very good predictor, just a single variant in recessive mode. The exome sequencing in the UK Biobank have also finally uncovered that gene in European populations, at least in white Britons. It is much rarer. It took about 50,000 exomes to discover it. It's only in 3 copies. So if you were to use that variant as a predictor in either population, one from the other, you would probably get it wrong. And so that's an example of how environment, selection, and many other factors can actually play into human phenotypes. So as a follow-up from this GHG paper, we started to look at empirical data sets of what might be underlying some of these differences. And I'm going to talk next about work we did in the NHGRI-funded population architecture using genomics and epidemiology consortium. And this is work led by Dr. Jen Wojcik, who is currently an assistant professor at Johns Hopkins University. And this was a consortium that was specifically set up to sample for diversity in the Americas to address questions not only for genomic discovery in diverse populations, but also understanding how genetic factors influence susceptibility to disease across populations. So the sampling in the study was approximately 17,000 Hispanic Latino, African American, 22,000 Hispanic Latino, about 4,000 Native Hawaiian and 4,000 East Asian Japanese ancestry participants, and then some others and some smaller portion of Native American participants. Okay, so that data was linked to over 200 traits and outcomes across both cohorts and biobanks. The paper that we published last summer in Nature focused on the first 26 of these, mainly cardiometabolic traits, some lifestyle traits, and 1 or 2 diseases, and did a GWAS. And despite the fact that almost every single one of these traits and diseases had been studied in much larger, usually tenfold, if not more, larger European ancestry populations at the time, we still managed to discover 26 novel loci that had not been previously discovered. And unsurprisingly, that's because those loci, the variants underlying those loci, index variants in the GWAS, were either rare or absent in European populations. But the other thing we were able to do was replicate about 1,400 known trait variance associations in diverse populations in PAGE. And this was a very nice data set that we could then use to look at replication across populations. So that's what we did, and we took the standardized GWAS, standardized effect size for associated traits in predominantly European populations from the GWAS catalog, and compare them to standardized effect size from PAGE analysis. We did this in a standardized way to control for sample size across studies. And sure enough, when you do that, and then you stratify the PAGE populations by the two biggest populations, African American and Hispanic Latino, you see that the effect size across all loci, and this is across now a plethora of traits, are attenuated in non-European populations. So in Hispanic Latino, attenuated even more so in African American populations. So what does this mean? We certainly don't think that this means that the biology of this trait are different across populations. In fact, we think the biology and maybe even the causal variants are mostly shared, but the estimates of effect size differ. So one of the other things that we could do using this data set is we could go through this toy example. Well, what happens if you use data from diverse populations with large studies of European populations? How does that impact your ability to implicate causal variants? And sort of under the assumption that if we had a better understanding of knowing what the causal variants are, we might be able to do a better job of robustly transferring risk across population. So in this exercise, we did a meta-analysis of height from the GIANT study using the 50,000 diverse participants in PAGE in one case, and then in blue using downsampled White Britain's 50,000 from the UK Biobank to do the apples-to-apples comparison. And in doing that for 390 loci associated in GIANT for height, we were able to estimate the posterior probability of the top ranked SNP in a credible SNP for each of those loci, and discovered that when you do that exercise, it is significantly higher in the GIANT data meta-analyzed with diverse populations than it is in GIANT data meta-analyzed with more European ancestry populations. And the reason for that is shown here in this exemplar, comparing those two analyses at one locus of association. And you can see in the middle layer here, the posterior probability in the European only analysis sort of fails to discriminate against 4 different SNPs. And the reason is they're all in tight LD in European populations, but does a great job of discriminating the credible SNP in single credible SNP when meta-analyzing with diverse populations. The reason is that LD breaks down in both African American and Hispanic Latino populations at this locus in a way it doesn't do in European populations. So now, what happens when we start to move from cohorts of 50,000 participants to cohorts of 500,000 participants, or soon in the future 5 million participants? How do these effects play out? So that has been of interest in my lab for many years, and particularly we think that aspects of recent demography are probably going to be very important for understanding massive scale human data sets. And we use identity by descent, which are long haplotypes that are shared with recent ancestors over time, to measure these at a population level to inform us about very recent demographic events, including bottlenecks, endogamy, migrations, and admixture. To do this at a scale of 500,000 people, and I'm going to show you some data from the UK Biobank, we had to actually reinvent the wheel and go back and build a method that would not tap out at 50,000, that would actually go all the way to 500,000. I'm not going to talk about it today. This work is up on bioRxiv, but we developed a tool for IBD detection called eyelash using locality sensitivity hashing, and this is work led by Ruhi Shimarani and Gillian Belbam with collaborators Christian Yu and Jose Luis Ambient. But one of the things that we did was really think about population structure and think about how this plays out at the more macro scale. So we went through an experiment where we took a data set controlled for population structure in the way that we traditionally think about this. So in this PCA plot, these are the 400,000 or so UK white, self-reported UK-born white Britons, also controlled for appearing within that blue meloid and PC space, so very densely congregating there. And this is commonly what we use to control for population structure when we're trying to factor that out as a variable in the types of association analysis we try to do. And I should say, what I'm going to show now is work led by Gillian Belbin, who's an assistant professor at Mount Sinai. So if you call IBD across the 300,000 or so of the biobank participants that live in that meloid, you get about 14 billion observations of pairwise sharing of haplotypes across those participants. If you link them back to regions in the UK, you can see that there is an average increase in shared IBD or length of IBD in more isolated parts of the country, for example, in the Outer Hebrides, in Shetlands, and in areas of Wales. But we can go a step further than that, and we can use methods that infer hidden structure within vast networks, and we can apply them to the vast network of IBD that we detect in the UK Biobank, and just allow, in an unsupervised way, around a community detector to randomly walk through this and assign communities where individuals in the community have more IBD sharing within the community than they do with individuals outside the community. And in doing so, we can detect up to 10 different distinct communities of IBD sharing. We can no longer use population labels to inform us what these different communities are, but we can use geocoding information again. So for example, the first community here tends to be enriched for people who are living in or where the ascertainment center is in central and west England. Likewise, this other green community here is enriched for people who are living in North Wales. Sometimes, however, geographic information is not really informative about communities, as we can see for this community, what we're calling community X here in purple. And this community is also very interesting because, as you can see on the y-axis, they are enriched for fraction degree sharing of IBD segments. In other words, we think this is evidence of this community not only being distinct, but also a founder population. And this is meaningful when we're thinking about how risk is transferable. So for example, if we take a PRS score for type 2 diabetes and we compare it in the main population compared to community X, we see a more subtle but significant mean shift in the distribution of the PRS score in community X. And this also translates to differences in risk prediction for this particular outcome, particularly in the upmost quartile of prediction, where we see a significant difference between the two groups. So what does this mean, and what's really going on with humans that make them so different for risk prediction compared to, for example, to domesticated animals? Well, some of the issues that we and many others these days are exploring are on the technical side. So thinking about using LD differences, particularly when you're using index SNPs rather than causal variants, but also issues of residual population stratification that tend to occur still in genome-wide studies, even when best practices are used to try and control for that. Even when we know the causal variant, allele frequency differences are still things that I think we need to be very careful about modeling across populations. And of course, background negative selection and positive selection are at play. And when you're starting to look across many, many, many diseases, this actually becomes more common than not. And of course, many and more complex things can be going on. And in fact, I think a large part of what's playing out here in our ability to predict is actually more connected to non-genetic factors that are probably linked to a lot of our very complex migratory history as a species and the environmental and societal factors at play. So just to finish up, I think it's a very exciting time for the field. And if we think about what the key driving questions are going to be for the next 5 or 10 years, I think we're really at the beginning of an era of using genomics data in a sort of whole-scale way and a broad way in health systems for risk prediction for preventive health over an individual's life course. So it's a single test that you can go back to many times. And the question will be how this plays out and how far we can move the needle with genomic information from treating and managing disease toward early detection and perhaps even intervention and prevention of disease. To do this, one of the very important things that has to happen is that we really have to accelerate transformation of genetic research into medical knowledge. This is generally something that happens, frankly, at a glacial pace. And to do that, I think we have to really tackle the biases in our current approaches and knowledge bases. Really what we want to be able to ultimately do is systematically identify at individuals, families, communities, or population at differential risk and understand the non-genetic factors that play over a patient's life. This is going to be absolutely critical to ensure genomic technology ameliorates, not exacerbates health disparities. So with that, I'm just going to finish up and acknowledge again the folks who led this work, including Gillian Balbin, Ruhi Shimarani, Alicia Martin, Jen Wojcik, longtime collaborator Christian Yu. Thanks to the funders and the many research participants who make this work possible. And the last bit pitch, if anybody's interested in doing this work and doing postdocs in New York. Thank you.
Great, thank you very much, Eimear. So to just time for one quick question. So the top voted one on Slido is, do you have any way to know if the attenuation of effect sizes in non-European populations is due to winner's curse in the GWAS or genetic background effects?
Yeah, so it's completely possible that winner's curse is at play here. The data I showed looked at 2 different non-European populations and showed that there was a different effect in different populations, which does suggest that it's not only winner's curse, that there is other factors at play in the attenuation.
Great. Well, thank you very much, Eimear, and I'm sure she'll be happy to participate in the Slack discussion as well. But we're going to have to move on now, and our next speaker in a pre-recorded talk is Anna Gloyn. Anna's a pioneer in functional genomics, focusing on type 2 diabetes. She's a professor within the Department of Pediatrics at Stanford, having recently moved from Oxford. And Anna's going to be talking to us about targeted and genome-wide cellular screens in pancreatic beta cells to aid type 2 diabetes GWAS interpretation. So.
Good morning to those of you on the West Coast. Good afternoon to those of you in Europe. Thank you, Matt, for the introduction. I would like to tell you a little bit about some of the work that we've been doing when I was in Oxford and more recently now that I've moved to Stanford on targeted and genome-wide cellular screens in pancreatic beta cells to aid type 2 diabetes GWAS interpretation. So we now know that bespoke studies on individual loci take many years, and our efforts in this space have largely been successful where we have had coding variants that are causal for the association and have given us a molecular signpost and heads up on which gene that we should be working on. But there are over 400 signals for type 2 diabetes, and the vast majority of these are not in coding sequence. And this means there's a concerted effort by the community to link variants to enhancer elements and to the transcripts that they regulate by asking a series of questions. Which SNPs alter which enhancers regulate which genes, and the SNPs that are associated with disease, do they cause a loss or a gain of function? And critically, we then need to ask the question, those transcripts that are identified, do they result in a disease-relevant phenotype when they are perturbed? All of these questions need to be asked in different contexts. For example, in the field of type 2 diabetes, we might be interested in knowing whether or not those variants are affecting regulatory annotation in the pancreatic pancreatic beta cell during development. We might also be interested in differences in regulation across diabetes-relevant tissues such as islets, liver, muscle, and fat. Also, it might be really important to study those regulatory annotations under different stimulations, because stimulation with glucose, for example, may unmask a regulatory annotation element that would otherwise not be seen. So how can we be confident when we've assembled these regulatory data sets and we have made links between variants to enhancers to transcripts, that these transcripts are actually disease relevant and that the perturbations are on the causal pathway to the disease that we are interested in, and not just pleiotrophy that's happening in the background? So one of the things that we need to start off by asking is, when we move into the laboratory setting, what model should we be using to study these relationships between transcripts and biological function? For type 2 diabetes, we've got pretty good evidence that we should be studying the pancreatic islet. We know a large number of the signals are working through that particular tissue. So we need a good cellular model for the pancreatic islet. Within that islet, a logical starting point to begin would be the pancreatic beta cell, which is one of the cell types within the pancreatic islet, but it's the one that's responsible for making and secreting the hormone insulin that lowers blood glucose levels. We know from the work of many in the field that there are important differences between rodent, and human islets and beta cells. And these differences are in things to do with the architecture of the islet, the genes that are involved in cell cycle regulation, the composition of ion channels, and also glucose transporters. So given these fundamental differences between a rodent and a human islet and beta cell, it seems really important to be modeling the genetic effects in a human pancreatic islet and beta cell. So what are our choices when it comes to working in this space? Well, for human primary islets, despite the fact that they weren't captured in GTEx and ENCODE in the original efforts, largely because these are not accessible tissues, they are available after you've processed the pancreas to isolate the islets from this tissue, and this can only be done easily on cadaveric donors. You can start to use this tissue for cis-EQTL mapping and also for putting down regulatory annotation. But it's worth noting that access to islets is limited. This is a heterogeneous source of material. There's variable function depending on how the islets have been recovered from the cadaver, and also how they've been isolated. And remember, it's a mixed cell population. So in 2011, there was a real change in the field, and an eagerly anticipated and awaited pancreatic human beta cell model became available thanks to the efforts of Raphael Schaafman, Philippe Rabichard and colleagues, who reported the EndoC beta H1 cell line. Since then, there have been a number of additional family members to this group that have got similarities and differences in how they have been derived and what you can do with them. We and others have put a lot of effort into characterizing this cell line and aligning it with a primary beta cell to see how well it measures up in terms of the key features that you would want within your cell model. Work by Benoit Hastoy, who was a postdoc working with myself and Patrick Roseman, was able to show that the electrical physiological properties of these human beta cells very much were aligned with primary beta cells, showing that they were a good model that you could use in the laboratory setting. However, they come at an expense, and that is that they are not necessarily the easiest cell line to work with. They are not a HeLa cell. They are not a HEC293 cell. They grow very slowly, doubling only once a week, making it very laborious to get up to the numbers of cells that you might want to work at scale. They also have complex media requirements, affinity, and can often die on you and then not work for months on an end. They require close monitoring and testing regularly to make sure that they retain their insulin secretory properties. A third model that could be suitable is a human iPSC or human ES stem cell derived beta cell. However, although these model systems are particularly good at modeling defects in pancreatic beta cell development, I think it's fair to say that although there has been substantial progress in the field, there isn't really yet a single protocol where we're absolutely confident that we're generating authentic, mature beta cells. In addition to that, we also have to take into account that making iPSC-derived beta cells is phenomenally expensive and perhaps a barrier to working at scale. So what do you need if you're going to think about the simultaneous analysis of disease-relevant cellular assays? Well, for example, you might be working either in a targeted or a genome-wide manner, going from hundreds to thousands of genes. You'll want to have a disease-relevant cellular phenotype that is scalable and, depending on the number of genes in question, can either be arrayed or can be performed in a pooled manner. Your cellular model, again, needs to be disease relevant, it needs to be scalable, and you need to have a model that you are able to genetically modify so that you can alter expression of the genes that you're interested in. So for this basis, we have focused the efforts in my laboratory on the human endoseed beta H1 cell line. So about 4 years ago now, we published our first work in this space where we took 300 genes at 75 GWAS loci for type 2 diabetes and for fasting glucose. And we looked at cellular phenotypes related to either the physiological stimulation of the cells with glucose or the pharmacological stimulation of the cells with elements such as tolbutamide or IBMX, and also looking at cell count. So across these 5 different cell phenotypes for 300 genes at 75 GWAS loci, all within this human pancreatic beta cell model. And this was work that was performed by Cern Thompson when he was a PhD student in the lab. And this experiment was possible because Cern worked with the target discovery group led by Daniel Ebner at Oxford to miniaturize the insulin secretion assays so that we were able to use acoustic liquid handling to get it down to 20 cents per data point. He took the genes that were located within 1 megabase of the type 2 diabetes GWAS signal and screened based on the fact they had to be expressed within an islet and also in a human primary beta cell. So taking these 300 genes at 75 type 2 diabetes GWAS loci, Cern identified 67 hits. 15 of these were for cell count and 52 were for one of those 4 insulin secretory phenotypes. A really important sanity check when we looked at our data was that there was enrichment for genes involved in monogenic forms of pancreatic beta cell dysfunction. And this was really encouraging and gave us confidence that the data that we were observing was meaningful. There were 45 genes involved in beta cell function at 37 of those 75 GWAS loci. One of the genes that we were particularly interested in was the transcription factor ZMIS1. Cern was able to replicate the findings using independent siRNAs. And the reason we were interested in this particular transcription factor was that an earlier study from our group performing cis-EQTL mapping in human primary islets, performed by Martin van der Bund, had identified a cis-EQTL that co-localized with the type 2 diabetes GWAS signal and resided above an islet-specific stretch enhancer. And when we knocked down the ZMIS transcription factor in a human primary beta cell, we were able to observe effects on insulin secretion. So here, using a second approach, we were picking up a similar story in terms of what the likely effector transcript was at this particular GWAS locus. So that was pretty good, 300 genes at 75 GWAS loci, but can we do any better? So we've turned our attention in recent years to trying to go genome-wide. And this has really been made possible by the work of a very talented PhD student in the lab, Antje Grootz. And what Antje has done is firstly develop a pipeline for performing CRISPR knockout in the human pancreatic beta cells. She's then been able to capitalize on this CRISPR pipeline to study its effect on insulin content. Now, insulin content, we decided, was a disease-relevant phenotype based on our work from the PAM gene, which is a type 2 diabetes risk signal. And we've been able to show that one of the primary ways that it has its effect on diabetes risk is through altering insulin content. And insulin content is a particularly attractive phenotype because, unlike insulin secretion, which needs to be measured in an arrayed form, this was amenable to a pooled approach. So Antia set out to establish an assay for measuring insulin content for a pooled screen. Now, this required a number of steps, including antibody testing, antibody specificity, fixation and permeabilization. And having spent about 2 years on this element of the screen, she then performed a proof of concept screen using a number of genes that we knew had or were likely to have roles in altering insulin content. And having established that the proof of principle was there, that we could detect effects on insulin content with this cellular phenotype, we moved into our genome-wide pooled CRISPR screen. We've worked with the Toronto Knockout Library, which has just over 18,000 genes with 4 guide RNAs per gene. Now, the tricky thing for us with this particular assay is the fact that we're working with this cell line that is very slow to grow. And that means it took us 4 months to generate the 700 million cells per screen that were required for this genome-wide pooled screen. So Antia really has been the driving force of this work, and at the most intense period was spending up to 12 hours a day passaging the cells that were required for the screen. So the screen itself included lentiviral transduction, puromycin selection, and then prior to the screen, we selected a reference population of the cells and then sorted on the basis of low or high insulin. Having collected the cells, we extracted DNA, and then there was PCR for the single guide RNAs and amplification, sequencing, and then, of course, we were able to map back the guide RNAs that we detected in the low and high populations of cells. Antia then moved on to do functional studies to validate some of her most promising hits. So to give you a summary of what the data that she's generated looks like and to give you some salient take-home messages. First of all, we were really pleased to see that there was really good correlation between the 2 replicate screens that she performed. We were also happy to see that the positive regulators of insulin content included genes that we were hoping to see, including insulin. One of the things we noted here was that insulin wasn't the top hit. So when we looked a little bit closer into this, we were able to see that that was because not all of the guide RNAs behaved consistently for insulin, and the top hits tended to be those where we saw similar behavior across the 4 guides. And that led us to think that perhaps what we should do here is be more stringent with our analysis and only include genes when we had more than 2 of the guide RNAs with a false discovery rate of less than 0.1. And in doing this, we ended up with 626 genes that we took forward into our analysis. So the first thing Antia did was a pathway analysis to see whether or not the genes that she was detecting that were altering insulin content made sense biologically. And in doing so, we were really pleased to see that type 2 diabetes was one of the pathways that was enriched, which was a terrific sanity check for us in terms of confidence with our data. So what were the first things that we started to think about doing with these data? Well, we thought it would be useful to go to a curated list of genes which had various degrees of confidence for being effector transcripts for type 2 diabetes loci. And this is possible through to a curated set of genes that have been uploaded into the Type 2 Diabetes Knowledge Portal by Anuba Mahajan and Mark McCarthy. Now, I'm not going to go into the algorithm that they have generated and used to rank genes with various levels of confidence for whether or not they are the effector transcripts at GWAS loci, but suffice to say there are genes with high degrees and are therefore called causal, some that have got strong degrees of evidence, and some that are moderate down to weak, etc. And the types of evidence that are going into this are things to do with coding variant associations, involvement in monogenic forms of diabetes, evidence for perturbation or regulatory annotation. So in looking at these data sets and going into those where there was moderate evidence, one gene that stood out to us was KCNK17. And when Antia looked in her data set, she was able to see that there was consistent performance of the guide RNAs, and that when she lost KCNK17 in her screen, she saw an increase in insulin content. As I mentioned, this is a gene at a type 2 diabetes GWAS locus, and there's data from a number of groups, including from Stephen Parker and Michael Stitzel, with work led by Arushi Varshni, that has shown that there are cis-EQTLs for KCNK17 at this locus. We also know that there is a coding variant signal in the neighboring gene and related ion channel KCNK16. So when Antia followed this up in the lab, although she was unable to replicate the effect on insulin content with independent siRNAs, she did observe effects on insulin secretion. And interestingly, when she knocked down KCNK17, she saw a reduction in the neighboring gene KCNK16 expression. So this is really interesting and adds to the complexity of what might be going on at this locus, where we have two genes with similar roles, they're related, and we have evidence for effects on their expression, and we have effects on coding variation. In Antia's screen, there is no evidence for an effect on KCNK16. We also looked at genes where there was only possible support for them being the affected transcript, and one that came to our attention was CalCoco2. And again, Antia saw very consistent behavior across the guide RNAs within her screen, and there was an association of gene loss with decreased insulin content. We've got a type 2 diabetes GWAS signal in this region. No strong biological evidence, but we do have a coding variant in CalCoco2 in the credible set at this GWAS signal. Antia moved into the lab to look into validating this, and having validated both the effects on insulin content with siRNAs in endo-CBTRH1 cells, she also observed effects on insulin secretion. In collaboration with Sung Kim here at Stanford, she's now established that CalCoco2 is expressed in human primary islets in both the alpha and beta cells, and she's recently performed CRISPR knockdown and is waiting to see whether or not she can replicate the effects on insulin content and secretion in primary tissue. So our screens here are really part of a much wider effort involving collaborators across the US and beyond, where we're trying to bring together various different screening efforts, both in cells and in model organisms, so that we can layer up data along with expression data and genetic data to come up with therapeutic hypotheses. These start genome-wide with our screens, not just on loss of function for insulin content, but we have a screen that will be imminently completed, where we've done the parallel effort for gain of function. We have insulin secretion assays in arrayed format, ER stress, perturb-seq in islets. We've been working with Marcel de Hood in Uppsala and with Sung Kim at Stanford to do screens in both zebrafish and Drosophila. And we're now adding to our repertoire of cellular models with Karen Mulkey, Susie Jacobs, and Melina Klausnitzer, so that we can bring in adipocytes, myocytes, and hepatocytes. We're then taking a smaller number of these genes into bespoke assays, similar to the ones that we've done for PAM and for SLC30A8, so that we can really drill down on these therapeutic hypotheses. So let me summarize. So I've shown you the efforts that we've gone to validate the models that we're using for our bespoke analysis and also for our screens. I've shared with you published data on a robust arrayed siRNA knockdown screen of 300 genes at 75 GWAS loci for 5 cellular phenotypes in a human pancreatic beta cell model. And I finished off with unpublished data where we've performed a genome-wide loss of function pooled CRISPR screen for insulin content in a human beta cell model. And I've started to show you how we're beginning to use our data to help inform on effector transcripts at type 2 diabetes GWAS loci. foci. So some take-home messages. Select your cellular model carefully. It needs to be disease relevant and authentic. You really do need to align multiple disease relevant cellular phenotypes. I didn't mention, but the concordance between the data that we did for the arrayed screen and for our genome-wide screen is only 20%, demonstrating that content is only one of a number of phenotypes that could be affected by our genes that are implicated in GWAS biology. We need to integrate genetic, genomic, and cellular data, and we need to do this across cellular systems so that we can really get the complete picture on what is going on. I'd like to wrap up by thanking the really talented Eimear and Cern, who are responsible for the targeted and genome-wide screens that I've shared with you today. We've been incredibly well supported by the Target Discovery Group at the University of Oxford. The high-throughput screening effort is led by Daniel Ebner, and we've been working with Elena and Robbie from his team. I'd like to give a shout out to my new colleagues at Stanford in ChemH, who are helping me with the ongoing screens, and my funders. And finally, I'd like to thank the rest of my team and the group of international collaborators that I'm working with, sharing data with, and bringing different data sets together to unlock the biology of type 2 diabetes GWAS. Thank you.
Thank you very much, Anna. That was a wonderful talk, and please keep the discussion going in the Slack channel. I'm sure there's a lot to discuss after that. So next up, we have Lude Franke. Lude is a statistical geneticist who's done a lot of work on the genetics of gene expression. He's a professor of functional genomics at the University of Groningen. And he's going to be talking to us today about the identification of key driver genes using single-cell trans-EQTL and gene regulatory network analysis. Over to you, Lude.
Well, thank you, Matt. Well, hello, everyone. First of all, I would like to thank the organizers for giving me the opportunity to present my work. And one of the questions that we would like to address is that we assume that many of the variants that are associated to a certain complex disease must be related in some way. And it's a little bit similar to these 10 seemingly unrelated places in the US. So let's assume there are 10 seemingly unrelated places in the US. Well, there's one thing that relates these 10 genes together, and that is the fact that they all happen to be the beginnings of rivers that feed into the Mississippi River. And the thickness of the line indicates the displacement of water. And I think this is one example of a literal downstream consequence. So what we aim to do for complex diseases is to do sort of the same. So let's assume a certain disease where only a limited number of variants have been found. If you take those variants, one of the possibilities that we have is to link them to genes through, for instance, cis-EQTL information. And maybe through trans-EQTLs, we observe that some of those variants are affecting some genes in cis, and that those genes happen to show a trans-EQTL effect on a converging gene called gene X. And maybe we see this for some of those variants in one tissue, whereas we observe that for other variants to happen in other tissues. And maybe we can observe that finally many of those variants ultimately also come together and elicit a strong pathogenic effect, causing ultimately disease. So is it possible to actually start identifying those important genes through different approaches? Well, one of the approaches that we have taken is to amass a large amount of blood EQTL cohorts through the EQTL Gen Consortium, in which we have been able to study over 31,000 blood samples. And we had three goals there. One is to identify cis-EQTLs. Another goal was to identify trans-EQTLs, where we initially had concentrated on 10,000 trait-associated variants. And the other goal that we had was to calculate polygenic risk scores for over 1,200 traits and to correlate each of these individual polygenic risk scores to gene expression levels, with also the aim to identify genes that are dependent on polygenic risk, their gene expression levels. So a few examples here that we observed were cancers. We observed multiple independent variants, for instance, associated to multiple myeloma, giving effects on specific genes, showing again a converging effect. And likewise, we observed the same for Hodgkin's lymphoma, where we observed that two independent variants were giving an effect on one particular gene, RTKN2. And for some other diseases, we don't see those converging effects yet, due to sample size issues, that these trans-EQTL effects happen to be often very weak, or due to the fact that these trans-EQTL effects happen to be also extremely cell type specific. But nevertheless, sometimes we do observe effects on trans genes in blood, on genes that are very relevant for the disease of interest. For instance, for testicular germ cell tumors, we observe that the associated variant is giving a cis-EQTL effect, but is also giving 3 trans-EQTL effects on genes that are well known to be involved in spermatogenesis or in male meiosis. And for other cancer-associated variants, we see similar pictures. A telling example, I believe, is systemic lupus erythematosus, where you can see that there are multiple independent variants on different chromosomes, all giving trans-EQTL effects on a very specific set of genes that are known to be involved in the type 1 interferon response. And we observe exactly the same when doing that through the polygenic risk score approach. And correlating the polygenic risk that we've calculated for SLE and associating that to the expression levels of every individual gene, we observe that an increased polygenic risk for SLE is positively correlated with increased expression of all these type 1 interferon genes. So indicating that through this approach, since none of the participants in our cohorts had actually SLE, it is already possible to identify some effects that are measurable in blood. So this is based on bulk data, and was one of the reasons that we also engaged in the single cell EQTL consortium. We've recently launched that initiative, and you can find the paper on eLife, and where we have similar goals. What we want to do is we want to harmonize the different blood EQTL cohorts that are now becoming available based on single cell data. We develop a harmonized strategy, making sure that cell type assignments are being performed in similar ways and that the data is being assessed and analyzed in the same manner, such that we can perform a federated EQTL analysis as we did for the EQTL Gen consortium as well. And what we aim to do there is to identify cell type specific EQTLs, but also, since many of these single cell datasets have been constructed also while using different perturbations, to identify cell type specific EQTLs, but also to identify context or response EQTLs. And finally, one of the goals is to exploit all the single cell data to develop personalized gene regulatory networks, on which I will touch upon later. So right now where we are is that we have developed standardized pipelines for single cell population genetic studies across different populations and cell type classification models that are robust across these cohorts. And in the next stage, we are going to conduct the first cis and trans EQTL analyses across multiple cohorts and are going to develop the first personalized gene regulatory networks, with the final goal to do this at a larger scale in 2022. So we've also used the single cell EQTL Gen consortium already, and that is in replicating the trans EQTLs that we had reported in the EQTL Gen manuscript. And what we observed there was that we can replicate not all of the trans EQTLs due to the fact that some of those trans EQTLs happen to be driven by cell type proportion differences. But what we observe is that among the ones that we can replicate, there's a very clear relationship between the amount of cells that you typically find in blood, due to the fact that when doing single cell analyses and PBMCs, you're going to get more single cell profiles for the more abundant cells as compared to the more rare cell types. But as you can see, there's a very clear relationship between the calculated genetic correlation between these trans EQTLs when taking the single cell data as compared to the bulk RNA data, and also that the relationship strongly is correlated to the amount of cells that is available. So changing gears a little bit is that we have used the expression data as well in order to study exome sequencing data recently. And for rare disorders, we in my department currently have a diagnostic yield of approximately 30%. And we reasoned that maybe by exploiting also a lot of gene expression data, we might be able to improve this diagnostic yield. And the way we approached this was to collect a lot of publicly available bulk RNA-seq data comprising many different tissues and many different cell types. And the idea was as follows, is that if we have a patient presenting with a clinician If we have a certain set of symptoms that this patient is suffering from, is it possible to take the genes that are known to cause that particular set of symptoms, and can we also, through co-expression information, infer additional genes that also might cause these symptoms if they are mutated? So let's suppose we have 12 genes that are known to be involved in unwinding DNA. If you then observe that there is another gene that is strongly co-expressed with all of those genes that are already known to be involved in this particular process, then you can also infer that it's quite likely that that gene is also playing the same role. And likewise, for symptoms, this is the approach that we took. So we made a website which you can find at genetwork.nl. There you can look up individual genes, you can look up these predicted gene functions and also these predicted disease symptoms. And you can also make networks out of sets of genes. But the most important thing is that you then can subsequently click on this button, which is called HPO gene prioritization, where you can type in a set of symptoms that your individual patient might have, such as abnormality of the face, neoplasm of the endocrine system, abnormality of the anterior pituitary, and intellectual disability. And then you can say, okay, now prioritize those genes that most likely explain each of the symptoms that this particular patient is suffering from. So when doing so, this is the list that you get. And you can see that there are a bunch of genes shown. On position 7 is the gene that is already known to cause this set of symptoms. This symptom is, telling for the Kleefstra syndrome. But you can see that there are another 6 genes that have an actually higher prioritization score. So what happens if you have a patient that is actually showing these symptoms, but you are unable to identify the mutation in the known gene? Maybe you could consider ascertaining the exome sequencing data of the other genes that are prioritizing even better than the known gene. So we thought, let's give this a try. Let's apply this to a set of real patients. So we took a set of 61 patients for whom we had conducted exome sequencing, but for whom we regretfully had not yet found the mutation explaining their disease symptoms. And of these 61 unsolved exome sequencing cases, we could then resolve another 15%. And we were able to identify a new gene that causes epidermolysis bullosa and cardiomyopathy. So that indicates to us that these approaches can also start helping us to improve this diagnostic yield. And I hope that by having a lot of other approaches combined, this is going to improve further also when taking other functional genomics data sets into account. So the next step that we take is actually that we start combining the information that I presented at the very beginning of my presentation and the exome sequencing approach that I showed you just a few minutes ago. One thing that we've recently observed is that if you take a GWAS, for instance, a GWAS on height, and you calculate gene p-values either through Pascal or Magma, and you take a gene regulatory network, what we then observe is that if you take the genes that are mapping within the established height loci, indicated here in green, and you take into consideration this gene regulatory network, then it turns out that the genes that are linking many of those genes inside the established height GWAS loci, that those genes that link them together all happen to be genes that are known to cause extreme forms of skeletal abnormalities when mutated. And through this approach, we can now start using GWAS information to inform ourselves through these gene regulatory networks on rare disease genes that also might play a role in particular cases. And if you contrast this, and this is shown on the right bottom of this figure, if you contrast that to just taking the genes that are showing significant associations inside the GWAS loci, you can see that AUC curve in dark gray. You can see that this approach is much better able to make inferences on those genes that cause extreme skeletal abnormality phenotypes. So what we hope is that by taking into consideration both GWAS information and accurate gene regulatory networks, we can start also now identifying new genes that might cause rare forms of these disorders. But that does require one thing, and that is that we need highly accurate cell type specific and also directed regulatory networks. So we can now link individual disease-associated variants, for instance, through various QTLs to individual genes. And you can do that both using cis and trans QTLs. But what we need is also ways and approaches to make these networks truly directed. And there are several approaches that are already available, but also new approaches that now can be used in order to start doing so. I give you one example here. One thing that we observed when doing eQTL analyses, in this case cis-eQTL analysis, was that we observed that certain variants were showing strong interaction effects. An example is shown here for a lipid SNP that is showing a strong cis-eQTL effect on FATS2. And we observed that SREPF2 is actually interacting with that eQTL strongly. So depending on the SREPF2 expression levels, you see that this eQTL effect becomes stronger or weaker. Well, SREPF2 is a transcription factor, the sterol binding transcription factor. And when you ascertain the ChIP-seq data that's available for SREPF2, you see that this transcription factor is actually binding on top of that lipid SNP that is having this cis-eQTL effect on FATS2. So what we believe is that it's the combination of both combined. We can identify an upstream regulator of FATS2 by exploiting this interaction effect, and it's that SREPF2 together with this particular SNP is eventually determining what the FATS2 expression levels will be. So you can also phrase it slightly different. In the bottom, you have an upstream and a downstream gene, and you anchor them using this particular SNP. So now let's assume that this downstream gene could also reflect the upstream effect of another interaction. And then you already get the idea is that by using and exploiting this interaction information that we can now start deducing from large eQTL datasets, we can also start making these networks directed. So this is something that we could establish using bulk RNA sequencing data, but this is also something that can be done exceptionally well using single-cell eQTL data. Here you see an eQTL effect that was based on single-cell data. It was based on information from only 45 donors, but due to the biological replicates that we had for each of our individual donors, we get a highly accurate picture like this. This type 1 diabetes SNP is giving a strong cis-eQTL effect on RPS26. But the interesting thing is that if you take different individuals that have different genotypes, you can see that the individuals with the TT genotype have a strong positive co-expression relationship between each other. So they are indicated on the top in green. There's a strong positive co-expression relationship between RPL32 and RPS26. And this is very much different from those individuals that have the CC genotype. They have a very strong negative co-expression relationship. And it indicates that by using a limited number of single-cell donors for your eQTL analysis, you can already establish relationships like this. So in order to actually replicate that finding, we used over 4,000 samples, indicating the added advantage of using single-cell approaches. So finally, what we are now going for is that we aim to also take advantage of the fact that we can identify those variants that also show a massive effect on the co-expression relationships that sometimes occur. So imagine that you would take the gene regulatory network from John, indicated in the left, and you would contrast it to the gene regulatory network from Kate. Then you can see that they are mostly similar, but there's one striking difference indicated in the red cross. And now let's suppose both of them have the same disease and there's a drug that's acting on this green gene. Well, due to the presence of the relationship of this green gene, this regulatory relationship on this orange gene, this drug will be effective for John and might help him cure his disease symptoms. Whereas for Kate, due to the absence of that relationship, this drug is still acting on this green gene, but it's not signaling its effect to the orange gene. So this is what we hope, is that by making such networks, making them cell type specific and making them directed, but also to make them personalized, we can start making much better inferences on how individual variants act, and also potentially at some point how this might be of use for also pharmacogenetics. So my conclusions are is that these functional genomics approaches can help to link variants to genes and ultimately to function. And I believe that by taking advantage of these new gene regulatory network approaches, is that we can link also different classes of genetic variation. We can potentially link complex disease genetics through regulatory networks to rare disease genetics. And I hope that by developing new experimental techniques and also computational approaches that leverage all this information in joint frameworks, we can improve substantially. So that is what I wanted to tell about, and I thank you a lot for your attention.
Great. Well, thank you very much for that, Lude. We've got time for one quick question, and that is from Rob Williams, who suggests this could be the perfect context for Bayesian causal modeling. Have you tried this?
No, we haven't tried that yet. Thanks for the suggestion. Yeah. Great.
Well, thank you very much, Lude, and well done for coping with your technical difficulties. So it came across very clearly. Thanks. So
thank you.
Finally, in our last talk of this session will be from David Kelley. David is trained as a computer scientist with a focus on genomics, and he's now principal investigator at Calico Life Sciences, where he focuses on investigating gene regulation in the context of aging. And today he's going to talk to us about machine learning to predict non-coding variant effects. So over to you, David.
Thanks so much for the opportunity to speak today. Very excited to experience the whole session, even if we're not in Copenhagen to do it. So I want to anchor the talk around an ICDA recommendation that I think is compelling, feasible, and I can best contribute to, which is to create this comprehensive human gene regulation map that really focuses on connecting distal regulatory elements to their target genes in a cell type specific fashion that can be used as a resource for understanding non-coding variants in these distal elements, as well as, it wouldn't hurt to make progress on promoter variants too. So I've been focused on this problem for a few years from some different angles. The main way that I've been able to make big progress, I think, is to frame a machine learning problem where I'm working with the many, many thousands of annotation tracks that we now have from the different consortiums to map regulatory activity across the genome with different assays listed here. So rather than studying these data as just a description, an annotation of the genome, I've been trying to set up a machine learning problem where you would give me the underlying DNA sequence of the genome only, and then I'm going to try to predict all of these regulatory attributes from that underlying sequence. And if I can do this well, then I have a strong argument that I'm able to read the non-coding genome to predict gene regulation. So the main tool that I'm going to use is called a convolutional neural network. These were really developed in image analysis and have become wildly popular in that space, but they also translate very well to biological sequence analysis as well. So the main structure that's interacting with the DNA sequence are called convolution filters. We're going to be scanning these across the sequence to recognize things like transcription factor binding motifs or nucleotide composition like CPG islands. And this is nice because these are little structures that bioinformatics has been using for a decade or more, or we've called them things like position weight matrices, something like that. So we're going to scan these across. They're going to tell you how well they recognize their pattern. These are all free parameters. You initialize them randomly. So you're going to learn all of these from your data. And then you're going to compute an activation function of the output, like a rectified linear unit, where you just zero out all the negative values. And then typically you perform an aggregation operation, like max pooling, where you're going to take the value at each place in this vector and take the maximum in a window. So by repeating this module multiple times, you get a deep network. So if the initial layer is able to learn these transcription factor binding sites, then the subsequent layers are stitching them together into a regulatory grammar in order to recognize the regulatory elements that we care about, the promoters, the enhancers, the insulators. So the initial attempts at using these were typically working with smaller sequences, maybe the size of a peak in some of these data sets. And while that worked quite well, I wanted to be able to tackle this, ICDA challenge of understanding long distance regulation. So I set up the model to work with much longer sequences. In this case, it's on the order of in this case, it's 130,000 nucleotides. So I'm always trying to push that longer. So after the convolution operations recognize the regulatory elements, I've tried a few different ways of then sort of sharing information across the sequence to be able to say, okay, if I'm a promoter here in the center, then if I can look around in a broad region, what do I need to recognize and care about? And the structure that I found to be most useful is called a dilated convolution, which generalizes the convolution operation by sort of hitting out farther with non-contiguous regions with gaps in the middle. And that's a kind of nice parameter efficient way to do it. Finally, by this time you've synthesized the nucleotide information into about 128 base pair bins, and then you're going to transform that to make predictions for any of the training data sets that you brought in. When I've been doing this lately, I've been working with on the order of 5,000 training data sets for the assays listed there. So you kind of split out the vectors to make predictions for all of these data sets in bins across the genome. So this works quite well in that you can train it on a set of chromosomes, most of the chromosomes, and then apply it to unseen chromosomes and see, qualitatively results like this, where we're able to recapitulate the data, reproduce the data from only the underlying sequence in most of the cases. Across the entire test set, the accuracy is competitive with the correlation between replicates. So the accuracy as measured by correlation is competitive with the correlation between two replicate experiments, indicating that that's sort of a soft ceiling on how well we'd be able to do, but also importantly pointing out that as the quality of the data, as the signal to noise in the data improves, then these models are expressive enough and they work well enough that you're able to train a better model. So this suggests that a second phase of this might be to look at some of these assays where the correlations were not as strong and redo them with newer technologies, newer techniques to further improve this sort of modeling framework. Often the most reliable way to continue to improve your machine learning model is to go out and get yourself more data. I was already training on basically the entire human genome, aside from some extremely repetitive elements. So it wasn't clear how to get it from humans, but we do have many other mammalian species, including the mouse, where we have lots of data. And so I kind of hypothesized that the regulatory programs between human and mouse, all of the transcription factors, their expressions in different cell types, and the motifs they bind are similar enough between these two species, which seem to be largely true from the literature, that you'd get better results for either species by training on data from both species together. And that turned out to be true. You can see that here on the right, where on the top, I trained a model on only human on the x-axis, or only mouse on the x-axis at the bottom. And on the y-axis then is a model trained on both of those species together and then tested on some held out sequences for CAGE, which is this gene expression profiling assay that focuses on the 5' end. And in both cases, you see that you get better results by training on both genomes as opposed to one, which potentially points towards a way that we could also continue to make these models better by going out and doing, encode for every animal in your local zoo or farm. The major reason to do this, to train these sorts of models, and there's all sorts of things you can do to understand gene regulation, to ask pointed questions. But once you've trained a model, you can give it any sequence. So if you're interested in genetic variants, then you can give it the 2 different versions of your variant, or more than 2. You can take the reference sequence and send it through the model. You can make an alternative haplotype and send it through the model. And then what you're going to get back is a profile across the local region for each of those alleles, which you can then compare. If you're interested in a particular gene, then you can slice out the prediction at that gene. If you're gene agnostic, then you can just take a sum across the entire sequence. So this means when you have a genetic variant you care about, send it through the model. If I trained on these 5,000 different human data sets, then I'm going to get back 5,000 statistics for how much that non-coding variant is predicted to influence activity across the region for each of those training data sets that I gave it. Here's an example of how you can put this to use. This is a variant that was fine mapped as a causal EQTL from GTEx whole blood. The first track here is the cage sample for PBMCs, which shows that the gene is very highly expressed. The reference prediction from the model is pretty good. It matches up with this track quite well. The third track then is taking the reference versus alternative allele and subtracting them. And so you see here that there's a little bit of predicted difference right around the variant. But for the most part, the difference comes at this transcription start site. So this is nice. The model has correctly learned that this variant influences transcription upstream at the transcription start site, which is about 35 kb away in this case, which is kind of about where we're at in terms of the distances that we can cover. You can go even further. So the score for the genetic variant is just modulating this, this nucleotide right here. Because we have this model, it's very fast. We can do an in silico mutagenesis, saturating the entire region around the variant. So we can take each position from the reference genome and introduce all of the alternative alleles and see the effect of each of those mutations, which is drawn in the heat map here at the bottom. And then from all of these mutations, you see the emergence of the motifs in this region. So you really get eyes into this regulatory element, this one maybe being an enhancer, that that go far beyond just staring at the reference sequence and along with a peak track where you really see the important nucleotides emerge, at least through the lens of this deep neural network. And in this case, it highlights this motif for a transcription factor called SP1, which is clearly the reason that this variant has a significant prediction. So I've performed a few different types of comparisons to EQTL data, which all show strong statistical correspondence. I'll highlight one performed in collaboration with Qingbo Wang and Hilary Finucane at the Broad Institute. So Hilary and Qingbo pointed out that in the latest round of GTEx, there are thousands of variants that have been fine mapped to a single very high probability causal variant, which can then be used to train a simple little machine learning model to discriminate between the very likely causal variants and a set of carefully chosen negative variants based on throwing the kitchen sink of variant annotations onto each of these variants. So we're going to annotate them with things like transcription start site distance, overlap with all sorts of peaks from all of these functional genomic data sets, and then tacking on these 5,000 features derived from comparing reference to alternative allele with the deep convolutional neural network. So then for each of the GTEx tissues, you have this set of causal variants, and you can train a random forest model to predict for each tissue in case there's significant differences between each one. And if we go into that random forest and ask which features it's considering the most, the distance to transcription start site is obviously a hugely important feature, but the scores, the predicted scores from my convolutional neural network, which are labeled Visenji here, are also very significant for getting good predictions with the random forest, which indicate that it's learning something useful here. So here's an example of how you can use that. Here is an EQTL association in whole blood, where if you go in and do the fine mapping exercise, you come up with 4 different variants here that are probably very linked. Looks like they have about equal probability of being the causal variant. The score from the random forest, which we call expression modifier score, EMS, really highlights just one of those 4 variants. And the main reason for that is because this genetic variant is influencing an SPI1 binding motif, which is quite relevant in the blood. So the prediction from this NG, the convolutional net for CAGE in different blood samples are quite high, and the EMS score then is quite high. So because the exercise of doing fine mapping with the population data alone, which is what you're seeing here at the top, where you're just working with genotype and phenotype matrix, phenotype being gene expression, and the EMS score, which is then based on all of these functional genomics data, they're orthogonal sources of information, and both of them are giving you back a probability. So it becomes then quite easy to combine those 2 things by treating the EMS score as a prior on how much you think each variant is likely to be an EQTL, and then letting the population data guide you to a posterior of which variant is likely to be the causal. So if we then go back to the GTEx data and redo the fine mapping exercise with the uniform prior on the y-axis here versus using the EMS prior on the x-axis, you see a shift towards smaller credible sets of variants. There's sort of a left shift in this table, indicating that in many cases, we're able to perform more effective fine mapping guided by the functional annotations. So I want to say a few words, since this conference is very forward-looking, about what's next. One of the things I'm very excited about is all of these genomics assays moving to single cell type resolution. So probably the easiest one so far to work with is the single cell ATAC-seq, which I've worked with this data set from the University of Washington, where they did about a dozen different tissues from the mouse, did a single cell ATAC-seq, and you end up with, in this case, about 85 different clusters of cells, which all represent different cell identities of some form from throughout the mouse. So you can then take these clusters, and although each individual single cell gives you a very sparse profile, if you have hundreds of them, you can easily then combine them into like a pseudo bulk profile and then feed them into the machine learning framework as individual tracks, as if they were bulk as before. And so, I've done this and it works quite well. In this case, then you're able to make predictions for how a genetic variant would influence ATAC-seq activity in these 85 different cell types. So you get a really nice zoom in from the tissue resolution to the single cell resolution. Of course, there's way more single cell RNA-seq going on. It's a little harder to work with RNA-seq. Everything I've shown you, the convolutional net is gene agnostic. You're literally just bringing in the coverage track and saying, have at it. And it's able to identify the promoters. With the RNA-seq, you have to play this game of, okay, I'm getting a measurement for a gene. Where's the gene? If I center on the known transcription start site of the gene, maybe I can get good results. So we're still playing around with how to do that as well as possible. I'm also very excited about these CRISPR screens that folks are doing to profile gene regulatory activity. I expect we will hear a little more about this in the conference, but I'll just sort of give the machine learning perspective. So the way these screens tend to work is you come in with a bunch of candidate enhancers, or maybe you come in
David,
totally unbiased.
David, any chance you can just wrap up in the next minute to give us some time for one question?
Yeah, absolutely. Okay. So, like I said, we'll talk about these later, so maybe I'll skip through. Basically, you're getting data about how different regulatory elements are influencing expression of the gene by directly perturbing. And I just want to point out, although it's hard to scale these things, this is great data for machine learning. We've been already using it for validation, and I think there's clever ways to use it as training data if we can get more of it. Okay, so with that, I hope I gave you an overview of how we can use machine learning models to predict regulatory activity to then go in and score genetic variants. Some of the things we're really working on next are working with even longer sequences so that we can do more with distal interactions. Like I said, we're making decent predictions at the order of tens of thousands of nucleotides, but we'd probably like to kick that up another 10x. And then, assays that sort of zoom in and give you more specific data, either with finer resolution or with single cell type resolution, are really exciting. But I think, the transformational improvements here are going to come with really clever experiments where we can go in and directly perturb these long distance regulatory interactions. So I'm hopeful that we'll have that. With that, I'd like to thank collaborators in human genetics at Harvard and Broad, as well as folks at Calico and DeepMind who are working with these deep nets with me. And thank you for your attention.
Great, thank you very much, David. That was excellent, and sorry for having to jump in there. So if maybe there's one very quick question about how does your work on the CNNs compare with methods such as DeepC?
Yes, it's very similar. We're both using the same sorts of machine learning algorithms. So, from there's 20 or so different design choices that you make about how you pre-process the data and how you exactly set up the convolutional net and how you train it and then what you do with it afterwards. And those are the places where there's like different tweaks and different decisions made by the different groups that are using these things, but the approach is quite similar overall.
Great, thank you very much, David. That was a very nice explanation for the non-specialist. So I'd just like to thank all of the speakers in this session for giving us a fantastic view of the near future and the more distant future and the diversity of the work that's going on. So thank you very much to you.